Good morning, Vanessa.
Good morning.
We're going to wait for a few minutes while other people come into the meeting. But if there are topics or questions that you'd like to post in the chat so we can start with them, please feel free. Okay, so my clock says 9 o'clock. So we can go ahead and get started and I'll add people in as they come. So I'm glad you could all be here today. And I hope that some of you have some questions that you would like to ask. This next lecture exam is covering 6 chapters. All 3 chapters on the circulatory system. We're talking about blood and the heart and the vessels. So in those, I mean, there are quite a few difficult topics such as blood typing and the pathway of blood through the hepatic portal system and the fetal circulatory system, plus lymphatic system, respiratory system, and a new one that's pretty complicated, the digestive system. So I'm ready to pull up any of the PowerPoints to show you images, to explain anything that you would like to hear. So just let me know. Where do you want me to start? It'll be a very boring review session if nobody has any questions. I have to put my foot up. I managed to break my toe last night.
So for the PowerPoint, I didn't see the jugular vein and the arteries. Was that something we were supposed to practice?
We have not gone through naming specific vessels yet. Other than, well, let me check. Some of the primary ones. Hold on, let me pull up an image.
So do you know where we should stop?
For the last one that's covered on the exam, you mean?
Yeah.
Yes, chapter 22.
Okay.
Just checking something quickly.
But we'll definitely go into detail later, right?
Yes, yes.
Okay.
Okay, let me share this with you. Hold on. Okay, so here you see an image of the specific large veins that you're responsible for. So we do have the external and internal jugular veins. These are the two major veins that drain the head. The superior and inferior vena cavas are the two veins that drain the upper body and lower body. Right and left subclavian veins drain the arms. And left and right common iliac veins right here drain the legs. So those ones you are responsible for knowing and identifying. And then for the arteries, we have these major arteries that you should know. So we have the ascending and descending aortas. So the ascending aorta is this short piece coming out of the heart before that rapid arch. Descending starts just here and goes all the way down into the pelvic cavity. So right and left common carotid arteries. These are the large vessels that are going up the neck, supplying oxygenated blood to the brain. We have the right and left subclavian arteries bringing oxygen to the arms, and the right and left common iliac arteries bringing oxygen to the legs. Those are the only ones I've asked you to specifically identify and be able to label up till now. In unit 4 of the lab, we're going to go over far more blood vessels.
Thank you.
You're welcome. There's a question in the chat. Could you go over fetal circulation of blood again? So this makes sense if you first understand the adult circulatory pattern. Let me pull it up. Give me a second. Okay, now let me share this with you. Okay, so here we see the pattern of blood in the fetus. The reason there's a different circulatory pattern in the fetus is because oxygen is no longer being picked up in the lungs. Fetuses obviously are not inhaling gas into their lungs. So where do they get oxygen? If you go back here to this structure, this is the placenta, and you see bright red, and the key shows that is highly oxygenated blood. So the oxygen is diffusing across the placenta. Now this vessel, because it's traveling to the heart, is going to be a vein. So again, we see a vein with oxygenated blood in it. So I told you, you want to look at the directions of the pathways as being more important rather than whether it's oxygenated or not. So oxygenated blood is traveling through the umbilical vein. So here's the umbilicus here, okay? And that connects the placenta to the fetus. And then you follow this pathway up and you get to a narrow connection between the umbilical vein and the inferior vena cava. Now, this connection is called the ductus venosus. So a ductus is just a narrow tube, and it's called venosus because it's connecting two veins. So the oxygenated blood joins the deoxygenated blood that's in the vena cava. So notice the mixing that occurs here. So now the blood is moderately oxygenated. And that is something you don't see in the adult circulatory system. You don't mix blood in the adult circulatory system. So the blood follows the typical pathway from the inferior vena cava into the right atrium. Now, normally, remember, blood will go to the right ventricle and then enter the pulmonary circuit. But we don't need a lot of blood to go through the pulmonary circuit because there's no oxygen to be gained in the lungs. So some of the blood in the right atrium is going to take a shortcut and bypass the lungs entirely. This shortcut is called the foramen ovale, which just means an oval-shaped hole. So that's this one right here. And it connects the right atrium to the left atrium. It's like a secret passage. So the blood will go directly from the right atrium over to the left atrium. So that is another unique structure. Then let's go back to the adult pathway. Right atrium to the right ventricle to the pulmonary trunk. Now notice we have a left pulmonary artery here and a right pulmonary artery here because you still need to bring oxygen to the developing lungs. So you still need to have blood flow to the lungs, but not that much blood needs to go there. So there's a second shortcut, and I'm indicating it here with my hand icon. And it is a short connection between the pulmonary trunk and the aorta. This is the ductus arteriosus. So remember, a duct is a small tube, and now it's connecting two arteries because the pulmonary trunk is an artery and the aorta is an artery. So this blood is going to bypass the lungs and go straight into the aorta. So notice all the mixing that takes place every time you merge these two blood streams. So what happens when the baby is born? Well, a lot of things have to happen very quickly. After the baby is born, the next thing that will happen is that the placenta will be passed as well. Basically, it needs to detach from the uterus, pass through the vagina, and be delivered with the baby. And doctors will now wait some period of time for this oxygenated blood to pass to the infant before they clamp off the umbilical cord. So the umbilical cord is no longer providing nutrients and oxygen to the new baby. So if you've ever watched a movie where a baby is being born, the first thing they always wait for is for the baby to cry. There's always this moment of, oh, will the baby breathe? Yes. So when you hear the baby cry, that implies that they've taken in oxygen. You can't scream without oxygen. So when the baby takes that first big breath, they inflate the lungs. And that inflation of the lungs allows gas exchange to start. So it's a really dramatic, rapid change that happens. So now that the lungs are providing oxygen, we need to undo everything that we've identified as unique about the fetal circulatory system and move toward the adult circulatory system. And we have to do it pretty quickly. So they clamp off the umbilicus and cut it. This lack of pressure causes the ductus venosus to collapse. It will heal closed and end up as just a little, almost like a skin tag, just a little piece of tissue. If you identify it in the adult through prosection, you will call it ligamentum venosum. Just a little piece of, looks like skin, and it will be embedded in the liver.
Professor,
Yes,
sorry to interrupt you, but I think we have someone waiting in the waiting room.
sure. Let me take a little break here and see if we can let them in then. Got two of them, thank you. Hi guys, we're just going over the fetal circulatory system. If you just joined us. All right. Okay, so ductus venosus collapses to make ligamentum venosum. That means that now the blood in the inferior vena cava is only deoxygenated, which is what we learned. So deoxygenated blood in the right atrium, we don't want it going to the left atrium, because if you do, you'll now be diluting rich oxygenated blood with deoxygenated blood. We don't want that. So that hole, the foramen ovale, closes. Now it doesn't dramatically collapse, it grows over, and this can take some time, but it will form a scar in the heart called the fossa ovalis. A fossa is a shallow indentation, so it will look like a little scar. This can take some time to close. And if babies show blue around the lips or other signs of poor oxygenation, it may be because this has not yet closed. But usually it does close on its own. Okay, the last one is the ductus arteriosus up here, which connects the pulmonary trunk to the aorta. Again, we don't want those two mixing anymore, because now the pulmonary trunk is filled with deoxygenated blood. Question? Okay, so you don't want the blood to mix. So because of changes in the pressure, basically blood no longer flows through this little duct. It doesn't collapse dramatically, but it does close very quickly. And it too leaves behind a little connection called the ligamentum arteriosum, but it's not open. And this is fairly easy to find if you do a prosection of a human or the dissection of an animal like a cat. You will see a little piece of tissue that links the aorta and the pulmonary trunk. But it's just a scar. It's basically showing what used to be there. All right. So this is where we get these heart defects that I included. These heart defects have to do largely with either congenital errors or errors of the fetal circulatory system that allow oxygenated and deoxygenated blood to mix. So patent foramen ovale, that is when the foramen ovale doesn't close. That causes the baby to get less oxygen to the cells, and you will see it as things like blue lips, also failure to thrive, low energy. Another fairly common defect is when the ductus arteriosus doesn't close. That's patent ductus arteriosus. And again, you will have a problem of blood flow going from the pulmonary trunk into the aorta. Now, these other ones are a little bit different. This ventricular septal defect means that the wall, the septum that divides the two ventricles is not complete. It's open. So blood from the right ventricle goes into the left ventricle. That is a very common defect. It just occurs due to how the heart was formed. It may require surgery. Depends how bad it is. I believe they usually do surgery. And then tetralogy of Fallot, I assume this is somebody's name. This is a series of defects. Basically, this is all of them all together. And so you have a real mixture of blood. And you may have a lot of malformations, such as a narrowing of the pulmonary trunk, opening of the septum, and the aorta may be in the wrong place. It's basically getting blood from both ventricles. So if that happens, the heart works harder and harder to get oxygen to the cells. And that causes hypertrophy. Hypertrophy means you've got larger size of cells, but it can lead to heart failure. So this tetralogy requires surgery to try and fix these defects and make sure that the blood entering the aorta is oxygenated. Okay. So that's the fetal circulatory system. Hopefully now you understand why those three structures are there, because they're basically solving a problem that fetuses have of how to move oxygen around when it's coming from the placenta rather than from the lungs.
Professor, I have a question.
Yes.
It's about the surfactant for the alveoli. Can you elaborate more about it? Like I had to look it over and understand what does it mean, like preventing from collapsing.
Yes. Okay. And that relates to what we just talked about. So let me just pull up that lecture real quick. That's from the respiratory system. And okay. All right. So that's the chapter on the respiratory system. So let me go down. Okay. That's talking about this right here. So if we look at the cells that you find in the lungs, we have one called a pneumocyte type 2 cell.
Right.
And you can see it labeled here. There's not a lot of them, but you have a few associated with each one of these sacs. Remember, that's an alveolus, one of these sacs. So what surfactant does is it coats this whole surface and means that the alveolus itself won't collapse. Because if it collapses, it can no longer carry out gas exchange.
Okay.
So surfactant is one of the important things that we see in the success of early premature infants. Because when you are born premature, they don't produce surfactant. And that means that when they exhale, parts of the lung collapse.
Okay.
Which is obviously going to make gas exchange difficult. So they have found ways to support the respiratory system of newborns who are preterm until they can make their own surfactant.
All right. Yeah, because I kind of got like questions about it, and I wasn't really too sure. I just wanted to make sure like I understood it.
Yeah, no, it's just a key part of allowing the lungs to remain constantly open so that each breath will fill the alveoli, and then you can carry out gas exchange. So without the surfactant, basically every time the baby breathes, it's almost like trying to take that first breath. So they have to work really hard to try and reinflate that alveoli that should have remained inflated.
All right. And this might be a little minor question, but I'm just wondering the difference between mucus and mucus. They're spelled differently. Like
Mucus and mucin.
no, like MUCUS, and then there's the mucus, MUCOUS.
Oh, I think that's American versus British.
Okay, are they the same though? Like,
Yes.
okay, all right, I got confused.
Yeah.
'Cause I put the caps on for when your videos and like it spelled it out, and I wasn't sure if you meant there's two different meanings.
Oh, no. If you're looking at the captions, it may have gone with one spelling. spelling is not my strongest point, so it's possible that I put the OUS. I think the OUS is the British form and the US is the American, but either one is fine.
Okay.
Good question, though.
And just out of curiosity, I just wanted to understand a little bit about the difference between the nodules and the nodes, the lymphatic nodules.
Ah, okay. Okay, let's pull that up. That's lymphatic system.
Because I know that nodules are a bigger version of the nodes.
Yeah, basically, oh, let somebody in. Okay, the nodes have a very distinct structure, and they are along the pathway that the lymph flows. And the nodules describe basically collections of tissues that are rich in lymphocytes.
Right.
So the lymph itself isn't necessarily all flowing through those nodules. Like the tonsils are really a nodule. They're a bunch of cells that have lots and lots of lymphocytes in them. And they're located in areas where they can detect and react to antigens.
Like the palatine tonsils.
Yes, right.
So the palatine tonsils will be an example of the nodules.
Right. So that's the idea. We've named them specifically, but that's basically what they are.
And so that would mean the nodes are basically where the is it a collection or?
Well, let me just show you. It might be helpful if let me just pull this up real quick. Okay, so we look at lymphatic system. Okay. Sorry, I have too many things open on my desktop. There we go. Okay. All right. So here's the lymphatic system. And if we take a look first at the lymph nodes, pull those up. Here we go. Okay. So notice the lymph nodes are in clusters and they're positioned along the pathway that lymph flows through. So it means if you look at a node up close, that lots of these tiny little capillaries, these lymphatic capillaries, will empty the lymph into a single lymph node. So we've got 1, 2, 3, 4 different lymphatic capillaries, and they're all dumping lymph into this node. The node is filled with cells and lots of lymphocytes. So this allows the body to basically filter through the lymph and look for anything that doesn't belong that can trigger the immune system.
Okay.
Okay, does that make sense? Okay, so those are the lymph nodes, and they have a very regular distribution. Notice each limb has its own cluster here, 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 and here. Okay, and you also have clusters coming back from the head. And then finally, there are clusters associated here with the digestive system. So anywhere that fluid is returning from gets a chance to be checked out by lymph nodes. All right, so that's a little bit different from these nodules, which are similar, but they don't have the flow of lymph passing through them. Instead, these nodules react to materials that contain antigens, but in this case, they could be coming in through the nose or something that you swallowed. So notice the orientation of these tonsils is associated with anywhere that a material could enter into the body through the mouth or the nose. So if you eat something or you get an infection in the mouth, that's going to produce antigens that the lymphocytes can react to. So if you've ever had strep throat, for example, that's an infection that starts in the mouth or in the throat, and that's going to cause swelling of the tonsils because they fill up with lymphocytes to attack that antigen.
That's why like it's swollen at times as well because they're fighting the bacteria.
Right. So the lymph nodes will also swell if they're fighting any kind of bacteria or virus or anything that they've identified as being an antigen. So when you're sick, right, the doctor will go and palpate your neck, and she's feeling for enlargement of the tonsils, but also enlargement of the lymph nodes, because if you notice these cervical nodes, they're also along the neck. So the cervical nodes react to antigens in the lymph, and the tonsils react to antigens that are in the throat or the mouth area.
Okay, that makes it more clear.
Good, good. And then there's other places where you find these nodules, particularly in the digestive system. There's a lot of these lymphatic nodules too. Okay. All right, so yeah, the body's just trying to create kind of ways of detecting and reacting to antigens, however they could be passing into or through the body. All right, so another question, are we supposed to learn the blood chart? Yes, you are. You need to understand basically who you can accept a blood donation from and who you could donate blood to. And it's based on the antigens and antibodies. So would people like me to go back over the blood?
Yes.
Yes. Okay, cool. That one is a tricky one. So I usually spend a lot of time on this in lecture. Let me close some of these. All right, so now I'm going to share with you, this is from the first chapter that's on this exam. This is chapter 17. Okay, and let's go down to where we talk about blood type. Okay, I think it's the first one. Yeah. All right, so first thing you have to understand is that we're looking at 3 different types of antigens. Now remember, an antigen is just something that can cause an immune response. Doesn't have to, but especially if it's an antigen that your body hasn't seen before, then the immune system will respond. Antigens, you're usually just talking about some kind of identifiable protein. Something that the body goes, wait, that's not mine, that doesn't belong here. And if an antigen is identified, then the immune system makes antibodies. Okay, so antigens trigger a response, and antibodies are made in that response. So it's important you keep the 2 separate. Okay, so where do we find the antigens that determine blood type? Well, they're found on the outside of a red blood cell. These are 2 of the 3 antigens. These are the first 2 that were identified, and they're called antigen A and antigen B. So in this diagram, they show antigen A as little circles and antigen B as little triangles. Okay, so let's say you have blood type A. That means on your red blood cells are antigens, antigen A. And because your body is used to that antigen, it will not mount any kind of an immune response to antigen A, no matter how it sees antigen A. If it's on your blood, it won't respond. If it's on the blood of somebody who donates to you, it won't respond. Because the body knows, hey, antigen A is fine, that's mine, I don't need to attack it. But if you put blood into the human body with antigen B on it, and you put that into somebody who normally is blood type A, then their immune system reacts to those antigens and says, whoa, wait a minute, that's not mine, and starts to make antibodies. Those antibodies will grab a hold. It's kind of like a lock and key. Grab a hold, the antibody with the antigen, and hold on to it. And it's holding on to it until another cell like a cytotoxic T cell, will come along and destroy the cell with the antigen. All right, so that's what the antibodies do. They make things clump up and stick together and be identified for destruction. So anybody who has blood type A has the potential to make antibodies to B. So this is the potential. It doesn't mean you have them. You will only already have the antibodies if your body has encountered antigen B previously. Does that make sense to people so far?
Yes.
Okay. All right. So if your blood type is A, you can get blood that has the antigen A. You can get a blood type that has no antigens at all, but you cannot get blood that has antigen B. Because if you do, your blood will clump, which will be very painful and potentially lethal. So the third antigen that was identified is called Rh factor. And to indicate if somebody has Rh factor, we call that positive. And if they don't have Rh factor, we call that negative. So if you look over here on this chart, you're going to see that throughout the world, blood type O is very common. O means no antigen A, no antigen B. Okay, but then depending on the populations, we'll have variations in A and in B. Okay, so notice that in some populations, such as Native South Americans, there is nobody in the population who has antigen A or antigen B. And we're not really sure why these differ from population to population, to be honest. There's some good theories, but I've never seen anything that really convinced me. Now, blood type AB means that you have both antigen A and antigen B. Now you look at the Rh factor, and most populations have Rh. Okay, so that means being positive is very, very common. There are some populations where people do not have Rh, and that means they would be blood type negative. All right, now that means we've identified here 8 blood types: O positive, O negative, A positive, A negative, B positive, B negative, AB positive, AB negative. Okay, that's 8 different blood types. Now, just to throw a wrench in it, I will tell you that there are other blood types. We're not going to go over them, but last thing I saw said there was over 20 different blood types. So these are by far the most common. So how do we identify what your blood type is? Well, when I was a kid, every single semester, it seemed like somebody would have us stab ourselves and bleed on a piece of paper to figure out our blood types. So they'd give you a lancet and you had to stab yourself and squeeze out 3 drops of blood. But now that we know that blood is rather hazardous, certainly after we figured out where HIV was transmitted from, we've pretty much stopped doing that unless they're really careful in the lab. But if you take something like Bio 1 class, they'll have you do an exercise that shows this same thing, even though it's with fake blood. Okay, so you squeeze out your blood and you have these 3 drops. And then you take a dropper full of what's called anti-A. And all anti-A means is that that liquid has antibody to A in it. So we've taken the antibody and just concentrated it. So when I put a drop of antibody A on blood, if that blood forms clumps, what does that tell me that was on that blood? What antigen was on that blood?
B.
Antibody A reacts to antigen A. A, right, antigen A. So remember, it's like a lock and a key. So the antibody A is going to look around and it only reacts if it finds antigen A. If there's no antigen A, no clumping. See, no clumping here, no clumping here. All right. This is blood type O, meaning there's no antigen A. This is blood type B, no antigen A. This is AB, so we get clumping. And blood type A, we get clumping. That clumping is called agglutination. All right. So the second bottle that you will be given is antibody B. You do the same thing, you apply it to the blood, and that's a different shaped antibody. Okay, it's not round like the previous one, it's pointy. And that will react only if it finds antigen B. So any clumping you see means that this blood has which antigen in it?
B.
B. This time we're looking for B. So we see agglutination, and that tells us this blood type has both the antigen for A and the antigen for B. Okay, and if your blood type is O, you have neither. If your blood type is B, you have no A, but you do have B. And if your blood type is A, then you have A, but no B. Okay, and then the third drop would be for the Rh factor. And agglutination would mean that you're positive. A lack of agglutination would mean you're negative. Okay, so with just 3 drops of blood and 3 different antibodies, you can tell which blood type you are. All right, so why is that important? Well, it's important because early on in medicine, people tried to save the lives of patients by giving them blood. And sometimes it was wonderful and it cured the person. And other times it made the person very sick. And sometimes it just flat out killed them.
Because it wasn't the same, huh?
Right. But at the time, they had no idea. And so it led to this general belief that you should never give people blood. And it turned out it was over 100 years before they figured out what the problem was. But there was generally a lack of interest in trying to use blood because it really wasn't understood what it was.
So if I'm a type A, for example, and
Yeah.
I give blood to a type B,
Yep.
that would result in an illness or possibility of death if I give too much of that blood, because the B would need antigen B, right? No. It would need antigen
No,
B.
your blood is blood type A, right?
Yeah. It's an example. I don't know my blood type, but like.
Well, don't worry about it. But let's say you're A and you give to somebody who's B.
Yeah.
So their body is going to go, A, that doesn't belong here. And the immune system is going to start to make antibodies.
So yeah, but if I give too much, it's gonna overrun the antibodies, or it will result.
No. It's not that. The antibodies aren't even made until the antigen appears.
Okay.
So remember, if you go back to your lecture on the lymphatic system,
Right.
and you see how the immune system works, remember it starts with the helper T cell,
Yeah.
and that will trigger a variety of things, including the plasmacytes, which are part of the B system. And now the plasmacytes make antibodies.
Right.
So as soon as I put your type A blood into someone who is blood type B, they will make antibodies. They don't have them yet. They will make them. And so it takes a while for that reaction to occur. So the person might ultimately get rather sick. But let's say you come back 2 days later and you give them more of your blood type A, because you're like, hey, they seem to have gotten better. They were a little sick, but now they look pretty good. Let's give them some more. Now the antibodies are already there,
Okay.
right? Now you're going to have a very rapid agglutination response. So what often happened was just that. The first time they got sick, they got over it. Second time they dropped dead, because those clumps would travel through, block the capillary beds, block gas exchange, and they would die painfully.
Okay, I understand that now.
Okay, good. So the antigens are what trigger the manufacture of antibodies. Here in the blood testing, we have the antibodies already made. So we see a fast response. But in the human body, if you've never been exposed to an antigen, it takes a while to start that reaction, right? Which is why when you get sick with a novel virus, it takes a while till you get better. All right, so that's the effect on donation and acceptance. So before I go on to hemolytic disease in the newborn, let's just look at, hold on, I have one more thing. This one right here. Okay, so this is something I would encourage you to try and fill out on your own. Okay, so first of all, for these 8 blood types, you have to figure out what kind of antigens are on that blood. So for example, if you are A negative, you have the antigen A. Okay, you don't have Rh and you don't have B. If you're AB positive, you have all 3 of them, right? What about if you're O negative? What antigens do you have?
Would it? None.
None. You don't have any.
That's exactly right. You don't have any antigens at all. And that is a great thing because it means that that blood put into anybody at all will not cause the immune system to react. So if you are blood type O negative and you have ever donated blood, you are receiving an awful lot of reminders from the blood donation people to come and give blood. That is the most valuable kind of blood they can get. And the reason is that can go into anybody without having to type and cross the blood. So if somebody's bleeding out right now, you can give them O negative. Right? So that is called the universal donor. Everybody loves O negative. Okay. Now the problem is if you are O negative, who can you get blood from?
Only O negative.
Only O negative.
Only O
That's
negative.
right. Go ahead. What was the question?
Oh no, I said only O negative.
Yeah. So if you go out here to donate to, an O negative can donate to all 8 blood types, but they can only get blood from O negative. All right. So let's look at the AB positive. We know they have antigens, all 3 antigens. And that means that no matter what kind of blood they get, they will never make antibodies. So the great thing about being AB positive is you can get blood from absolutely everybody. You're easy. No matter what they give you, you're fine. But who can you give blood to?
Only yourself, AB positive.
AB positive. So we don't really need your blood being donated very badly. It's not something that they're looking for especially, okay, because you are the universal recipient. But as a donor, you're not so useful. All right. So I would urge you to try and go through all of this to straighten it out in your own mind. Remember that makes antibodies. This is saying if exposed to an antigen, can you make antibodies? So if you have the antigen A, you're never going to make antibodies to A. But if you're exposed to blood type B, you will make antibodies. So you will make antibodies to B, and you will also make antibodies to Rh because you don't have that antigen naturally. Okay, does that make sense to people?
Yes.
Okay, I know there's a lot going on here, so it's a topic I like to go over when possible. Now, the last thing is hemolytic disease of the newborn. And this is something that was noticed by doctors after they understood blood typing, because you would have a woman who was Rh negative, and she would have a healthy baby. But then after that first baby, she'd have repeated miscarriages. She wouldn't have a second successful pregnancy. And they couldn't figure out what the problem was because it wasn't every pregnancy, and they couldn't predict who would have problems and who wouldn't. but they did notice it happened in Rh negative women. So what happens, we now know, is that during birth, the mother is exposed to the blood of the newborn. Childbirth is a messy business. And so there is some exposure between the mother and the child. So the mother, if her first baby was Rh positive, the mother would be exposed to a new antigen, meaning that she would make the antibody. Not a lot of it, but some of it. And remember, after being exposed to an antigen, you make memory B and memory T cells. Those memory B cells mean that the next time you're exposed to that antigen, you're going to start making antibodies really fast, right? That's what gives you immunity. But in subsequent pregnancies, if she had a pregnancy with another baby who was Rh positive, her own immune system would attack the pregnancy, and it would result in stillbirth or miscarriage. So these days, it is very typical right after birth to give a woman a shot that will stop her from making antibodies to Rh. It's especially important if she's negative and her husband or the father is positive, because then you have a likelihood of having a baby who is Rh positive. And just that simple treatment at the time of birth has stopped most of the problems of the hemolytic disease of the newborn. But sometimes they will still have to treat a newborn who does have some difficulty related to this immune system issue. All right? All right, so let's see what else. Somebody asked, will there be the same number of questions for each chapter like last exam? Yes, there will. I believe I've got it currently set up. I haven't completed the exam, but I think right now I have 8 questions per chapter. There's 6 chapters, and then I'm going to have some labeling, some short answers, matching, things like that. So it should be very similar to the previous exam, but because we have more chapters, there's fewer questions per chapter. Okay, would we have to label the digestive system? No, you would not. No, I don't think, if you check the review sheet, I can't think of any labeling that goes with the digestive system, no. So I'm not going to show you a picture and ask you to label it. Labeling would be more for those major blood vessels I covered, for the heart. I don't think there's any other labeling. Those are the 2 big ones, I think. Blood vessels and heart. How did you guys do with the digestive system? Did you focus on those things I told you, like where does the body first digest particular molecules?
Yes.
Okay, so where does the body first digest carbohydrates?
It starts in the mouth with the amylase, because amylase helps digest carbohydrates. It's a digestive enzyme, right?
It is very good. And the specific molecule it digests is starch.
And starch is basically it's the molecule of glucose, right? Basically.
Yeah, you just stick a whole bunch of glucoses together. And so amylase will break the bonds holding the glucose together. And that is chemical digestion. And that is amylase. And you'll know it's an enzyme because it'll end in the letters ASE. Okay, where does the body first digest proteins?
That in the stomach.
Stomach, yeah, I was say stomach.
Stomach, yes. Okay, so the acidic environment of the stomach breaks the bonds that holds the protein in a very tight, elaborate 3-dimensional shape. So think about a big blob, and you can't break down the individual amino acids while it's all tucked into this big structure. So by breaking the hydrogen bonds that hold that structure together, it opens up the bonds between the amino acids. So you can start to break those down. And the enzymes to break down the amino acids, that's largely in the next section in the duodenum. But the breaking of those hydrogen bonds to expose the amino acid bonds is the beginning of the digestion of protein. So that is the stomach. Okay, good. And then the last one are the fats. So where do we start to digest fats?
It's in the small intestine,
Which part of the small intestine?
the duodenum.
Good. Okay, the duodenum. So this relies on this process called emulsification. And so we know that fats and liquids don't mix, or fats and waters, I should say. So all throughout the digestive system so far, the fats have only been exposed to water-based liquids. So emulsification happens with bile. And basically what happens is that you take big chunks of fat and you turn them into little tiny pieces of fat. They're still surrounded by water. Okay, so when you have emulsification, the fats and the waters, they don't mix, but the fats are now so small that it's possible to attack them on their surface area. So the bile emulsifies the fats, and then the lipase is the major digestive enzyme that will break down the bonds holding the molecules of the fat together. And where does lipase come from? Where is it made?
Hit the pancreas.
Very good, the pancreas. So the pancreas is absolutely key to producing these digestive enzymes. Basically, you cannot digest your food without the pancreas. And this is why it's so dangerous if the pancreas gets plugged up at those little fine ducts that empty the pancreatic juices into the duodenum. If that gets blocked, you get pancreatitis, which is incredibly painful. And essentially, the pancreas starts to digest itself. And it cannot release the enzymes that are necessary to digest the food in the digestive system. Okay. So yeah, pancreas is an absolute key player to chemical digestion. Okay, good. Let's see, is there anything else you'd like me to go over? I know the digestive system has a lot of stuff going on in it. So if there's any questions that you want to ask about that, please feel free.
Can you go over the 4 tunics of the abdominal tract? Like, where are they located exactly throughout the digestive system? I'm kind of confused on that.
Yeah, let's take a look at that. All right.
The mucosa, submucosa.
Yeah, that stuff, exactly. Okay, so let's go to where those are. All right, so here are the tunics. So essentially, if you just treat the digestive system like a tube from mouth to anus, you're going to find these 4 layers or tracts throughout. So you're going to find it in the esophagus, you're going to find it in the stomach, large intestine, small intestine. Okay, so we're not talking about any of the accessory organs, just the tube-like structure that the food passes through. Okay, so we're going to go from deep to superficial. And so the deepest layer is called the mucosa. And that means that all of these structures here are within this layer. So it's not like a single tissue layer or anything like this. There are multiple structures within the mucosa. And so these are just the major players within the mucosa. But essentially, you're going to have a layer of epithelium, right? Because every lumen has to be lined with epithelium. Could be very, very thin, right? The simple epithelium, or could be stratified depending on the conditions inside that part of the tube. Then you have this layer called the lamina propria, and that doesn't change much from area to area. This is where a lot of the vessels will pass through. Remember, epithelium doesn't have blood vessels. So this is the connective tissue that you find just deep to the epithelium. And then you have 2 fairly thin layers of smooth muscle. Okay, Then you get to the submucosa. So this is the next layer after the mucosa, and you can see it's labeled here. This is, again, lots of connective tissue, lots of the vessels passing through on their way to the mucosa. And because we have in the mucosa some muscles, you have to have nerves to control those muscles. So the submucosal nerve plexus is this delicate layer here you can see is a yellow sort of series of crossing structures. These are the nerves, and those nerves are going to pass action potentials to the deeper muscles in the mucosa. All right, the third layer, this is where the real muscle is. This is the muscularis externa. So when I talk about the layers for peristalsis and segmentation, we're really talking here about the muscularis externa. And these are normally 2 layers, except when you look in the stomach. And then the deepest layer is going to be circumferential. So it goes around the lumen. And then the more superficial layer here, that's longitudinal. So it runs the length of the tube. And these 2 together are the ones that carry out peristalsis and segmentation. And between the 2 layers, you see another nerve plexus. So plexus is just sort of a branching layer of nerves. All right, so 1 plexus for the deep layer and 1 plexus for this more superficial grouping of smooth muscles. Okay, and these also include the sphincters, and sphincters are the circumferential muscles that control diameter. So the sphincter can be contracted so nothing can pass through, or it can be relaxed so that food can pass through the digestive system. And then the last layer is the outermost layer, and it can be one or the other. If it's adventitia, this is a connective tissue with lots of collagen fibers, so it's strong, and it connects this tube to structures around it. So a lot of times inside the body, you will find these layers that help to adhere one structure to another so they're not moving around, or worse yet, twisting or turning in such a way to interfere with the blood flow. Because if you cut the blood flow off, then that part of the body will die. On the other hand, this outermost layer can also be called serosa, which means this is serous membrane. Remember, we find serous membrane in either the thoracic cavity or the abdominal cavity. So the outermost tunic will be serosa when we're looking at something like the small intestine or the large intestine. And that's when we get into the different mesenteries. Remember, mesenteries are made up of serous membrane. Adventitia, you would find, say, if you were looking at the outermost layer of the pharynx, for example. So when you're way up here in the pharynx, okay, that's obviously not in the thoracic cavity. So the connections between the pharynx and surrounding structures, that would be known as adventitia. All right. Is there anything else you wanted to go over with those tunics?
No, that was good. Thank you.
You're welcome. So everywhere I went through the digestive system, I had little notes about this might be different. So the Roman numerals refer to, actually, I think I switched them from being Roman numerals. So when you go down later and it talks about how layers are different, I try to refer back to those. So the mucosa, for example. Okay, so this is number 1. So this is the mucosa. What is different about the mucosa in the stomach? Well, one thing that's different is it has these gastric pits. So instead of just having a layer of epithelium that's either stratified or simple, you have these infoldings, these deep pits. Okay, so I try and kind of tie that back to the original tunics of the digestive system to help you figure out what I'm looking at.
Sweet. Thank you.
You're welcome. Okay. Other questions?
And then I had another question, kind of similar to what we just went over, but
Yeah.
but this is going to be like in the jejunum and the duodenum.
Yep.
Like how to identify the microvilli from the villi and from the plica.
That's a great question. Okay, good. Let's go back there. the digestive system. Okay. So we're looking. Here's a good place to start. Okay. All right, so focus for a moment down here on the lower right. Here we see the duodenum, the jejunum, and the ileum. Okay, these are the three different parts of the small intestine. And there's some structures that you find. We've got these plica, which are folds. We have villi, and we have microvilli. So usually I draw this out for you. It's a little easier that way, but let me see if I've got another image that I can. Let's go to this one. Okay, so first thing are the plica. The plica, this whole big extension is the plica. And so if you see how there's lots of folds, and if you think about it, the food is going down the tube and the plica is at a 90-degree angle to the direction that the food is going. So think of these like speed bumps. So the more plica, the slower the food has to move as it kind of bumps its way down this tract. So the plicas are going to slow the passage of food, because one thing, if we want to get all the nutrients out of the food, we don't want it going through quickly. Everybody's had an unpleasant experience of food poisoning, and you know what happens when food goes through your digestive system quickly. It is not well digested. Okay, so that's the plica. All right, and so that shows these here. So where do you have the most plica in the small intestine?
Jejunum?
Yes, the jejunum. Okay, that's where you have, see how the submucosa comes up quite substantially. And that submucosa is what raises the surface of the lining and creates those plica or folds. Okay, so we can already see how the jejunum is trying to slow that food down. And that makes sense because the jejunum is where the majority of nutrient absorption takes place. The other thing these folds do, in addition to slowing down the food, is that they help to create more surface area. This physical property of surface area is really the emphasis of all of these different folds and extensions. The more surface area you have, the more area for nutrient absorption you have. Okay, so the plica are going right across in opposition to the flow of the food, like little speed bumps. Okay, now look closely at the plica, and you're going to see that it's covered with these folds. Those folds are called villi, which means fingers. So all along these speed bumps are millions of tiny little fingers. And if you zoom in on one individual villi or finger, here we go on this image. So here's a close-up of a villi. So singular is villus. And if you zoom on in, you're going to see that each of these villus structures is covered with cells, of course, and these cells have microvilli. So if villi are fingers, microvilli are teeny tiny little fingers. And you should remember the microvilli because we studied them before and they're a part of the cell membrane. They're caused by folding the cell membrane. So microvilli are folds. They're on top of villi, which are folds. The villi are on top of plica, which are folds. So you have tiny folds on bigger folds on bigger folds. And all of that gives you more and more and more surface area. Okay, so here, this again is one villus. And so you see many, many villi here, and they are on the surface of an individual plica. So if you go back to comparing the different layers, you can see that you have the biggest plica with the biggest villi in the jejunum. The duodenum, we do have the plica. Here's a nice bump right there. The villi are a little smaller. And then when you get down to the ileum, instead of plica, now we see nodules, lymphatic nodules. So remember, these are filled with lymphocytes. So by the time we pass into the ileum, the villi are smaller, the plica are not present. You still have microvilli, okay? But there's less of an emphasis on absorption and more of an emphasis now on reacting to antigens that might remain in the non-absorbed material. So whatever's left after your body's absorbed the good stuff, some of it is going to trigger the immune system. There might be things in that food that the body will consider to be antigens. Okay, so here's another way of looking at it. Okay, so this whole big thing that comes up, okay, and it's part of the submucosa. This whole thing is one plica. The little fingers that you see all over it, each one of those is a villus. And if you zoom in on one villus, you're going to see that they are covered with cells that have microvilli on them, which we learned a long time ago is about absorption. Okay, so you just want to maximize absorption in the jejunum. And as you move to the ileum, there's less of a functional emphasis on absorption. And you start seeing those Peyer's patches, and that's what we call these nodules here. So these Peyer's patches are found throughout the digestive system, but they're most common in the ileum and into the large intestine. Okay. Did
Thank
that
you.
help clarify?
Big time. Thank you.
Good. Okay. Any questions? It doesn't have to be a deep, serious question. It could just be something you're not fully comprehending, something you want me to clarify. Let's see. Do you guys understand the hepatic portal system?
Sort of.
Sort of. Okay, well, it doesn't hurt to take a look at that one because I find that that can be kind of confusing. Is that, let's see, oops, that's digestive. I don't want that. Okay. That
Hey,
is,
Professor.
yes.
Hey, I'm trying to think about how to phrase this question because, like, I know that you can't like tell us like, hey, this is going to be these are going to be the exact questions that are going to be on the exam.
Nope,
But-
can't tell you that.
Yeah, I was curious though. Are you able to tell us like which concepts, I guess, are going to be like more? You're going to have more like matching and what's the other one? Labeling questions.
Well, the best way to understand that would be to look at the review sheets and look out for anything that says be able to label. In terms of
okay.
matching, no, I don't really have anything. Usually matching is for categories when there's a bunch of things like the different cells that you find in the gastric pits, right? So when you have 3 or 4 of something and they all make something different, or like on the last exam, I think I had it for the bone cells, because you had 4 different bone cells and they all did different things. So that kind of thing lends itself to matching.
Okay, thank you.
You're welcome. All right, so I found the image. Let's take a look now. So this is from the chapter on vessels. Okay, so here's the hepatic portal system. Hepatic also always refers to the liver. A portal system is when you have an additional capillary bed. So normally we saw that blood goes from artery, capillary, vein, back to the heart. Okay, and then you had exchange taking place in the capillary bed. With a portal system, and we're going to see another one. This is the first of 2. With a portal system, you have an extra set of capillaries. So let's start to see where this vein begins. So we have multiple veins, and notice they're all coming off the digestive system. So any vein that's coming off the digestive system is going to have a lot of those nutrients that we just went through all that effort to absorb. So around the stomach, around the small intestine, large intestine. So all of these are nutrient rich, but it's deoxygenated, right? Because we've already carried out gas exchange. But the absorbed nutrients could contain toxins, especially toxins that can affect the brain. The body goes through a great deal of effort to protect the brain from toxins. We'll see again about something called the blood-brain barrier, which is a second way the body tries to keep toxins out of the brain. But the detoxification takes place largely in the liver. So one of the big functions of the liver is to remove toxins and to break them down. So all of this deoxygenated blood that is rich in nutrients is going to travel to the liver. But we have to get those nutrients out of the blood and into the liver. And that requires a second capillary bed. So this vein right here that's going into the liver is the hepatic portal vein. And notice it branches into smaller and smaller veins. And then at the end of these veins is going to be a second capillary bed. That is where nutrients can leave the circulatory system and enter the liver. So the liver can break down toxins. Then flowing away from the liver, it's a little hard to see up here, but it's called the hepatic vein. And that is draining the liver. So any nutrients that make it past the liver will re-enter the capillary bed and travel away from the liver via the hepatic vein, and then go into the inferior vena cava and off to the right side of the heart. So this detoxification function of the liver helps you to understand why, if somebody overdoses either intentionally or unintentionally, usually you're going to see it in the liver. So for example, I've heard that people who try to overdose on acetaminophen, the painkiller, right? They can have their stomach pumped and recover just to die a few days later. Because although they've removed much of the toxin, the remaining absorbed toxin goes straight to the liver and unfortunately destroys the liver. And you cannot survive without your liver. Okay, so that's what the hepatic portal vein is about. Any other questions that people want to ask? You all understand the B and T cells? It's another one that's another kind of multiple versions of cells that have different functions.
Professor, I have a quick question about that.
Yeah.
You said in the lecture that B and T cells mature differently. Can you elaborate on that a little bit?
Okay, so basically the reason they're called T cells is because they mature in the thymus. The thymus produces chemicals that are required for the maturation of the T cells. The B for B cells means that they mature in the normal place, which is in the bone marrow. So especially when you're young, the thymus is very important for the development of those T cells. So there is an image. Let's see, hold on, let me pull it up. Okay. Right one. No, I'm looking at the wrong lecture. Hold on. I need the lymphatic lecture. Okay, here we go. Oh, this image right here. Okay, so the red bone marrow is where mitosis takes place of, all the cells that are going to make up your blood. So we have stem cells, and a stem cell is just a cell that can undergo mitosis and make more than one type of cell. All of the cells that are found in your blood can trace back to a single type of stem cell called a pluripotential stem cell. But from that can arise two basic types of stem cells. One of them is the lymphoid stem cell. Okay, so all lymphocytes come from that lymphoid stem cell. The other one was the myeloid stem cell. And the myeloid stem cell we saw on the image about the blood, I believe, and that gives rise to all the remaining blood cells. But the lymphoid stem cells make the lymphocytes. Some of the products from these lymphoid stem cells are going to travel to the thymus. And this gland produces hormones. The hormones allow these to mature into T cells. The remainder of the products of the lymphoid stem cells are going to stay in the red bone marrow and become either B cells or NK cells. So you really have these three different groups that derive from the lymphoid stem cells, and only the T cells require the thymus to mature.
Okay, thank you.
You're welcome, Samantha. Are there questions?
I just wanted to elaborate again. I'm still trying to copy down what you said about the hepatic system. So if you overdose on a drug, and you pump out the drug in the stomach, you can still die a couple days later if it affects the liver.
Yeah, that happens with acetaminophen, I believe. That it's really sad because often people have been saved from overdosing, and most of the time they're really happy that they didn't die, and so people can be rejoicing that they averted an overdose, just to have the person die a few days later because their liver fails. So do not overdose on acetaminophen. It's nasty stuff. It really destroys the liver. And inadvertent, like think about, it's very common to have liver damage for people who abuse alcohol, because alcohol is a toxin. It's broken down in the liver. If you drink moderately, your liver can keep up with it. But if you drink a lot, then your liver starts to be unable to detoxify the alcohol, and the liver is replaced instead by either fat or scar tissue, which no longer detoxifies the alcohol. And if you lose enough of the liver, then again, you go into liver failure and you die without a transplant.
What's the drug called again?
Acetaminophen, I believe, is the one. So acetaminophen is the name for Tylenol, I think. They're painkillers. There's ibuprofen and acetaminophen, and one of them is worse than the other, and I think it's the acetaminophen. But basically, people who attempt overdosing on painkillers can find themselves in a tremendous amount of difficulty, even if they survive the attempt, because of the liver damage.
Because of the toxin that they have, right?
Yeah, well, it's toxic to the liver, destroys the liver. Yeah, in small doses, it's fine. Again, the liver can break it down, and it's not a problem. But in overdose situations, even some people who take large doses regularly can experience liver failure as a result. So you have to be careful with them. Even though they're over the counter, they can be toxic, toxic enough to destroy the liver.
Okay, got it. Thank you.
You're welcome.
One more thing, I just wanted to elaborate.
Yeah.
The one that I question about is the ulcer. Mentioned the ulcer you mentioned in the digestive system. You said if you don't guard the lining of your stomach with mucus, you're gonna start in your own stomach wall because it's made out of the proteins.
Sorry, you cut out. I got you. The last word I heard was proteins.
Oh, if you don't guard the lining of your stomach with mucus,
You'll digest it, yes.
okay, so that would basically lead to an ulcer, which would be a sore.
Yeah, it can lead to an ulcer, a bleeding ulcer, or even a perforated ulcer. So essentially, that mucus lining keeps the contents of the gastric pit away from the stomach itself. So we know now that most ulcers are caused actually by bacteria. And I don't know the exact mechanism, but that bacteria must damage the mucus lining sufficiently to allow the digestive enzymes and chemicals to start acting on the stomach. So ulcers are very painful, and we long associated them with diet and stress. And although those things can suppress your immune system and exacerbate the problem, can't talk today, we now know that Helicobacter is a bacteria that can cause an ulcer to occur. And the guy who discovered it, actually, nobody believed him. They thought it was ridiculous because it was generally accepted that it was diet and stress that caused ulcers. So the guy proved it by drinking basically a big glass of Helicobacter and then documenting the ulcer that he developed, and then taking antibiotics to destroy that same bacteria. He recovered from the symptoms of the ulcer. Most scientists are not willing to hurt themselves to prove a point, but some are. And after that, we now know that, checking to see which bacteria are living in the stomach is an important part of treating an ulcer.
What was the treatment again?
Helicobacter. Let's see if I can spell it in the chat. Helicobacter. That doesn't look quite right, but it's something like that. Let me find it.
Yeah, because I had to, when you mentioned stuff, I had to do my research on it, and I had to make sure like it was correct of the same direction you're making.
Let's see, it's Helicobacter pylori. Put it in the chat. But it really changed everybody's understanding of what an ulcer was. There you go. So yeah, you can read up on it. It was really interesting. And I think it's an interesting example of how people are resistant to changes, even though doctors are supposedly scientists and they understand the scientific method. People still didn't want to think that it had anything to do with bacterial infection. It's a lot easier to blame people's lifestyle choices and, that they're not taking care of themselves or they're drinking too much coffee, things like that. But it turns out that's not really the big problem. It's mostly to do with this particular bacteria.
So that's why we need mucus to help prevent that.
Sorry, can you speak up?
That's what we need the mucus to help prevent something like that from happening.
Right. Because once you start to damage the lining below the mucus, then very rapidly you're going to start getting blood because it's going to get into that submucosal layer. And then eventually it is possible to eat all the way through the stomach wall. And that is almost always deadly, because as soon as the stomach content starts spilling out of the stomach, you're going to start digesting everything. So it's incredibly painful. And it would cause an emergency situation, but it's very likely that there would be a lot of symptoms before that ever happened, because just getting through the wall of the stomach is very painful, and you have to go through all those muscles. There'd be bleeding, all kinds of things. Yeah. All right. Well, if nobody else has any questions, I'll hang out here until you leave, but it doesn't look like anybody else has any. I will be posting this. Your quizzes should be open, so you should be able to take your lab quiz and your lecture quiz for week 10. And then your exam should open during your Zoom time. So you can ask any questions before then, sending me emails. If you have a later Zoom time, of course, I have my my Confer Zoom hours on Tuesday or Wednesday. So if you're a Wednesday or Thursday class, you can still come in and ask me questions during those times. Okay. So you can take off if you want to. I don't think we're going to cover anything else unless anyone's being shy. I'm really glad you guys came. I hoped it was helpful. Let me know if you have any more questions, of course.
Thank you.
Thank you. Stay safe, stay healthy.
Thank you, Professor.
You're very welcome. Have a lovely weekend, Vanessa.
You too.
Thanks.
So, because I'm writing it down on my flashcards.
Mhm.
So it makes a hole through the stomach that will digest the stomach itself eventually.
Yeah, basically, the hole is created by the digestive enzymes and the acid in the stomach. So it's just the mucus is protective. So if you disrupt that mucus, now you've exposed the lining of the stomach. And since we already said this, the stomach is where you break down proteins, and well, your cells are pretty much built of proteins, you're going to start to break those down.
Okay, got it. All right. Huh. And for lipase, that's how it's called.
Lipase. Yeah.
Is that an example of like produces enzymes that help digest our food? or-
No, the lipase is an enzyme. So if
Oh.
it ends in ASE, it's an enzyme, and it's specifically to digest lipids.
Okay.
Yeah. So the pancreas is going to make all kinds of enzymes that are going to break down remaining carbohydrates, proteins, and fats.
Okay, for sure. And emulsification is basically breaking big pieces of fat to make into little pieces of fat.
Right. Not to be too disgusting, but emulsification is how you make mayonnaise. So
Yeah, I was like, wait, you mentioned that.
yeah, you can just, if you just take oil and eggs, they don't mix, right? Because one's water-based and one's oil-based. But if you beat the snot out of them, you'll break them into small enough pieces that each tiny droplet of oil will be surrounded by water. And that's going to stop the oil from reforming into large pieces or droplets of oil. So if you think about it, bile is sort of turning the fat into the digestive equivalent of mayonnaise. It's kind of gross, but that's how it works.
All right, for sure.
So without bile, you cannot digest large amounts of fat.
Oh, I had one question that I kind of got confused on. Like,
Yeah.
it got me overthinking, but so
Okay.
I'm just thinking otherwise for the liver, like, you know how if it's basically a filter in a way.
Sort of.
Yeah.
Yeah.
So if let's say if I were to take a painkiller just to stop pain or an allergy pill,
Mhm.
how would it get to the liver, or would it go? How would it go to the liver to filter it out? Or
Everything goes to the liver through that hepatic portal system.
really?
Mhm.
Oh,
Everything absorbed by in the stomach, small intestine, large intestine. Now, we do absorb some things in other areas, like you absorb through your mouth, through your stomach, certain amounts, and it depends on the chemical. But the majority of what you eat is digested and absorbed through the intestines, and all of that is going to pass through the liver.
because of the would it be because of the vessels of the
The
capillary?
hepatic portal vein is specifically carrying the absorbed nutrients to the liver to make sure that you minimize the exposure of the body, especially the brain, to potential toxins. Because the liver can break down most toxins. And once broken down, they can be safe. But the brain cannot it cannot break these down. So the brain could be destroyed by a toxin.
Yeah, because I was just curiosity, like I thought about it, and I wanted to elaborate, like how would it get there, and
Yeah. Now
'cause
you can get drugs other ways. Obviously, like there are medications that you take under the tongue.
Yeah.
So those are going to enter into the circulatory system. It's going to take a while for them to hit the liver.
Right.
So there are ways to introduce toxins to the blood that don't go directly to the liver. We're focusing on digestion here. So most of the digested materials will make it to the liver before entering into general circulation.
Right. So basically all of them will enter through the vessels in to get to the liver to the basically the veins.
Yeah, so if you go back to that image I showed you of the hepatic portal system,
Right.
you're going to see the veins running. They're traveling away from the small intestine, away from the large intestine, away from the stomach. They all merge together as the hepatic portal vein and enter the liver. And the capillary beds in the liver are going to allow materials in the blood to go from the blood to the actual liver tissue, where they can be broken down by specialized cells in the liver.
Okay. All right. Okay. Yeah. Okay. Yeah. All into the living system.
And then, Professor,
Yes, Matthew.
I had a question for the pericardium on chapter 18 for the heart.
Okay. Yep.
On one of the slides, on the slide of pericardium, there's fibrous pericardium and then serous pericardium.
Yeah, let me pull that up. I know which one you're talking about.
Okay.
There it is. Okay, this one right here. Right, so these are the different layers.
Mhm.
Okay, so the fibrous pericardium, this is a tough, thick layer, and it physically anchors the heart to the surrounding structures, including the diaphragm and the blood vessels. So basically, if you've ever seen like open heart surgery or images of the heart while it's beating, it's very active. It's, it's like jumping around in the chest. So you have to sort of hold it in place so that it's not creating a lot of friction and smacking against everything else around it. So this strong fibrous pericardium is an anchor. But just deep to that fibrous layer, see that little thin white layer there? That is the parietal pericardium. So it's just deep to the fibrous pericardium.
Okay.
Okay, so the serous pericardium is just talking about the parietal and the visceral pericardium. Those are thin layers. We know between the two is where we're going to get the serous fluid.
Mhm.
The pericardial sac includes this thick fibrous layer, and then just the parietal pericardium here.
Okay, that's what I was like. Okay, so it's just the parietal pericardium, not the visceral.
Yes, right, right. So if you dissect a heart, often when you get it, there will be a tough sac around it. Like when we would do the cat dissections, you would have to cut through that sac and open it, and then you would see the heart.
Mhm.
And on the surface of the heart, sort of vacuum sucked against the surface of the heart, that would be the visceral pericardium. You cannot remove that easily. That doesn't just pop off.
Uh-huh.
But the fibrous pericardium and the parietal pericardium, those are together in that outer sac, the pericardial sac.
Got it.
Okay. So if you've ever heard of people having like fluid in the pericardial sac, what's called a pericardial tamponade, okay, that would be if fluid accumulates in this space between the two serous membranes. But they can insert a needle through those and remove fluid that accumulates there if necessary.
Okay, thank you. That made
You're welcome.
more sense.
You said fluid in the
You can get fluid trapped between the pericardial sac and the heart. And if that happens, it creates pressure pushing on the heart, and it can ultimately stop the heart from beating. Called a tamponade. Sorry, tamponade is a dish you make with olives. Tamponade with an M.
What chapter is this on?
Oh, the image that we're looking at here, this is from the heart. So this is from chapter 18. Tamponades are not in your notes. I was just expanding.
I'm writing it for EMT, so I will know this like when I know what they're talking about.
Okay. So
That's
yeah, you can get fluid due to, say, infection. You can get fluid accumulating deep to the pericardial sac.
So fluid in the pericardial sac.
Yeah. That's going to be a tamponade. But I don't know that EMTs will be, hopefully they're not going to ask you to drain a tamponade. I think
Oh, no,
that's pretty
I-
serious.
I just want to know this so I can be ready, like, for this. So
Okay.
fluid in the pericardial sac, and they have to drain through a needle, huh?
Yeah. That's how I've seen it done. Again, not a doctor,
Yeah.
but I'm always interested in this stuff. When any kind of medical condition relates to an anatomical structure, then I'm interested.
And this could what again? This could stop your heart from beating?
Yeah. Yeah. Because you're putting pressure on the heart from the outside. So the heart's going to be working harder and harder, and that can lead to heart failure or just failure of the heart to be able to move enough blood at all.
Oh, speaking of pressure, can you elaborate on I mean, the volume increase and the
Respiration.
respiration, yeah.
Yeah, you know what? I'm glad you asked about that. I'm sorry most people are gone, but it'll be in the recording because that seems like something that people have gotten a little bit confused about.
I
So
started
let's-
I understand it, like, but I just wanted to understand a little bit more, like what.
Well, it's more physics than anything else, this particular part. Okay, so, all right, here's the idea here. So when you contract the external intercostals,
Right.
that raises up the sternum and it elevates the ribs. So when you do that, you take an area, a volume, and you make it bigger. But it turns out there's a relationship between pressure and volume. So if volume increases, the pressure inside that area will decrease.
Okay.
Air or blood or anything else will move from an area where where pressure is high to where pressure is low. So the air outside your body is going to move into the lungs because the pressure is lower in the lungs than it is outside the lungs.
And by pressure, do you mean like the pressure it is to take a deep breath, or for the lungs?
No, I mean the pressure created by the atoms that occupy that space.
Okay.
So if you imagine we have a volume here and it's filled with atoms, and I suddenly increase the volume, I don't add any atoms to it, I just make a larger space. The atoms are going to move farther apart from one another to fill that space. They're going to bump into each other less. So the pressure inside that space has gone down. The
Oh,
other way is true too. If I take a volume and I decrease it, I'm now pushing the atoms closer together, creating high pressure.
Okay.
So exhalation is really about decreasing the volume, which causes the pressure to go up, and then the air moves from high to low pressure. So you just have to think about sort of what is the cause and what is the effect. The cause is muscle contraction changing volume. The effect is that pressure is altered and air moves in or out of the body.
okay. All right, I'm getting it now.
Good. Yeah, we feel like we suck air in when we inhale. But actually, we just change the volume, which changes the pressure, which pulls the air inward because the air is moving to an area of less pressure.
Would an example be like if you had a balloon and you just squeezed it, you're creating less volume, which happens to be more pressure?
Right. And then the air pushes out of the balloon faster if there's a hole.
Yeah.
Yeah. It's just a basic physics.
Like Boyle's law, right?
Yeah. Yeah. The volume relates to pressure inversely. Yeah. But if people haven't had physics or, they just don't see that connection, it's helpful to think about it that way.
Yeah, I was lucky that I'm taking chem right now as well. And so
Good.
we're going through all like the gas laws and everything. And this happened to be at the same time. So I was
And you
like,
went, aha, that's how it works.
yeah.
Yes. So people keep going, well, the external intercostals make you breathe. Well, they do, that's sort of the effect.
Right.
But what they're actually doing is changing volume.
Yeah.
Yeah.
All right, I think I'm good with all the questions I had for this
Good, Matthew.
session. Thank you for helping. Really do
Oh.
appreciate it.
Anytime. It's my pleasure.
So you have a great rest of your weekend, and I'll see you. We'll talk again next Monday.
Okay, that sounds good. You have a nice weekend too.
All right, thank you.
You're welcome.
Wait, are we taking an exam this Monday?
What, Brian?
Are we taking an exam this Monday?
Are you in class on Monday? Is that your day for Zoom?
I thought we take an exam this Monday, don't we?
There's an exam your next Zoom day.
Oh, I was like, oh, see, this Zoom day. I'm like, wait a minute, are you taking it? Oh.
He's on a different Zoom day. That's all.
Oh, okay.
Yeah. Don't freak yourself out. Whatever your Zoom day is, that's the day you have your exam.
All right. I think that's it for me.
Good.
Oh my gosh, it's a lot of flashcards.
Well,
just-
that's good. That's
Well, I
good.
barely finished it yesterday, actually at 12 a.m. today, I think.
Wow. You just got through going through the lectures, huh?
Yeah, well, I when I make flashcards, I make sure I get every information. So I ask, like, get every information I can so I can understand it more clearly.
Mhm.
And that's probably why I missed my Zoom meeting with you on Tuesday, because I've been staying up so late almost every day, and I was like, it's 11 a.m. I thought I had something to do. Oh,
Yeah, well, I don't take it personally. I hang around in the Zoom room for about 7-8 minutes, and if you don't show up, I just shut it down. So it's not the end of the world. If you want to make another meeting, that's okay.
yeah.
Just make sure you set your alarm clock.
Well, I did. And I still slept over it.
Yeah, I got a couple of kids like that. They're staying up late, trying to catch up to their work, and then they oversleep in the morning. So
Well,
I understand.
Because I close when I work, so I'm there till 11:30, and like I don't
Mm.
sleep till like 5 a.m. or 6 a.m. And then, like I said, I'll wake up in 15 minutes. That 15 minutes turn to 6 hours later. I'm like, damn it.
Yeah, you got to have time to rest. Your brain needs that time to detoxify. It turns out your brain is cleaning itself while you're asleep.
Oh yeah, I felt that yesterday.
Yeah. So
I
we get stupid without sleep, definitely.
I had to meditate, then I fell asleep. All right. But I think that does it for me.
If you have more questions, you can send them via email. I'll be
Yeah.
checking my email.
Oh, wait, one more question. It's
Okay.
about peristalsis and segmentation.
Yes,
Peristalsis is basically how the bolus moves through your digestive system,
yes,
and it's like the rhythmic contraction, like
yes.
really
And
speaking.
it pushes the bolus forward. Yeah.
Yeah, and this includes the circumferential and longitudinal layer, which would be the muscularis externa.
Yes, exactly.
And for the segmentation, what does that basically do? I know it squeezes.
It's mixing.
Oh, it's a mix.
It's basically when you want to mix the bolus with something like digestive enzymes from the pancreas.
Okay. All right.
So it's just more of a back and forth, moving the bolus back and forth and breaking it into smaller and smaller pieces so that whatever you're trying to mix it with can come into contact with the surface area.
Okay. All right.
And so that involves the circumferential muscles really instead of the longitudinal muscles.
All right. And one more question for the pancreas. I kind of get confused, but so what does it produce again? In the main pancreatic duct or sphincter, like
Okay, well, it's producing, basically we talk about pancreatic juices.
Okay.
So one of the things it has to do is neutralize the chyme.
Okay.
So remember that everything coming out of the stomach is very acidic.
Right, right.
And so we are no longer focusing on breaking down large proteins. So we need to neutralize that acid and bring the pH level back up towards neutral.
Because chyme is usually low.
It comes out, yeah, chyme comes out at about pH of 2.
All right.
And so you got to bring it back up towards 7.
all right.
So
and-
that's one function of the pancreas. The second function is to produce digestive enzymes, such as lipase.
Okay. And so when we have that mixed with bile,
Yes.
that's basically what will help break down all our whatever we digest in our system.
Well, no, we're just focusing on things that haven't been digested yet. So by the time you get to the duodenum, we've started digesting carbohydrates. We've started breaking down large proteins into long strings of amino acid.
Okay.
Now we need to do two things. We need to emulsify fats, and that's what the bile will do. And we need enzymes to break the bonds holding the amino acids together, holding the simple sugars together, and holding the building blocks of the lipids together. So all those digestive enzymes are coming from the pancreas.
Okay. All right. Okay. I'm getting it now. That makes more sense.
Good.
All right. That does it for me. Thank you, Professor. Really helped a lot.
You're welcome anytime.
I'm glad I'm awake.
I am too. You would have missed a lot if you'd slept through this one.
Oh yeah, I know. All right. Have a good one.
Thanks, Brian. You too.
Bye.
Bye.